You woke up this morning, and you were older than you were yesterday. Not by much, not enough to see. But somewhere in your body a cell is dividing right now, and as it divides, it is making a tiny mistake. It is forgetting, just slightly, who it is supposed to be. In Texas, a naked mole rat is about to turn 31. It will not get cancer, it will not get arthritis, it will give birth in its third decade. Its DNA looks almost exactly like it did when it was 2. In the cold dark off the coast of Greenland, there is a shark swimming that was alive when Beethoven was a child. The oldest one ever recorded was 392 years old. Greenland sharks do not reach sexual maturity until their 156th birthday. In a research lab in Pomona, California, biologists have been tracking the same hydra for 41 continuous years. There is no detectable senescence. It is not slower. It is not less fertile. It is the same. And then there's us. By the time you turn 40, your eyes are already starting to fail. By 50, your hearing. By 60, your bones. By 80, half the people you love will be gone. Every species on this planet has spent millions of years adapting to staying alive. Whales solved it. Mole rats solved it. Sharks solved it. A jellyfish off the coast of Italy, when stressed, simply reverts to a polyp and starts its life over. We didn't. And here's what's strange. We know almost exactly why we age. We have the framework. We have the genes. We have mice in laboratories that we can age and then partially unage again. $3 billion seeded the largest biotech launch in history specifically to crack this. There is a man in Los Angeles who spends $2 million a year on his own body, hoping he will not be the last generation to die. So why hasn't anyone actually stopped it? The answer is stranger than fiction. It involves more than 200,000 missing Japanese centenarians, a Harvard professor resigning his own academy in disgrace, a jar of his own father's plasma, and a single, quiet paper that suggests the most famous evidence for living to 110 is just pension fraud. For most of the 20th century, biologists assumed aging was wear and tear. Like a car, parts wore down, cells got damaged. The free radical theory dominated for decades, the idea that oxygen-related rust slowly built up inside you until your body simply gave out. But in January of 2023, a team led by the Spanish biochemist Carlos López-Otín published a paper in the journal Cell that has now become the operating framework of the entire field. They identified 12 interlocking failure modes of aging: telomere attrition, genomic instability, mitochondrial dysfunction, cellular senescence, where cells refuse to die and instead drip inflammation into the tissues around them, stem cell exhaustion, chronic inflammation, the slow shift in your gut bacteria, and at the center of all of them, the strangest one of all, epigenetic alterations. Your DNA is not what changes when you age. Your DNA is, for the most part, the same molecule you were born with. What changes is which genes get switched on and off. Imagine the DNA in every one of your cells as a piano. The keys do not change. The instrument does not break. What changes is the sheet music. As you age, the sheet music gets blurry. Skin cells start playing notes meant for liver cells. Brain cells start playing nothing at all. This is the information theory of aging. It says you are not wearing out. You are being slowly forgotten by your own cells. And there is one more piece of damage running alongside it. In 1912, a French chemist named Louis-Camille Maillard described the reaction that browns toast and sears a steak. The same chemistry happens much more slowly inside you. Every time your blood sugar spikes, glucose binds to the long-lived proteins in your body, your collagen, your hemoglobin, the lens of your eye, where the proteins are the same ones you were born with. They are never replaced. They cannot be. Which is why, by your 7th decade, the inside of your eye begins to brown like the crust of a roast. The byproducts are called advanced glycation end products. They cross-link your tissues. They stiffen your arteries. They accumulate in the plaques of Alzheimer's brains, in the wrinkles of sun-damaged skin, and in the cataracts of your grandmother's vision. You are slow cooking. You have been slow cooking since you were born. Every spike adds another layer. The lunch you ate in 3rd grade is still chemistry locked into proteins your body has not replaced and never will. So if we know all of this, the failing piano, the slow cooking, the silent rust, why can't we stop it? In 2023, a Harvard biologist named David Sinclair tried to answer that question by doing something strange. He tried to prove the theory of aging by inducing it, by making a young mouse old, on purpose, without changing a single letter of its DNA. His team built what they called the ICE mouse, inducible changes to the epigenome. They engineered a way to make tiny cuts in the DNA in the regions that do not code for anything. Not to introduce mutations, just to send the cell's repair crew running, over and over, the way an X-ray, a long flight, a sleepless night, or a single drink quietly sends yours running inside you right now. Within 6 months, the mice were unmistakably old, hunched backs, gray and thinning fur, reduced grip strength, They forgot how to navigate mazes. Their epigenetic clocks ran a full year ahead of their actual age. A student in the lab once texted Sinclair a photo of one of them with the message, "We have a sick mouse." Sinclair texted back, "That's not a sick mouse. That's an old mouse. Same DNA, same genome, just older." Then his team turned on 3 genes, OCT4, SOX2, and KLF4. 3 of the 4 Yamanaka factors, the molecular switches that can turn an adult cell back into a stem cell. They deliberately left out the 4th because the 4th one causes cancer. The mice partially reversed. Their epigenetic clocks ran backward. Their tissues began to look young again. In a related experiment, the same 3 factors had previously restored vision to blind mice, a 5-fold increase in optic nerve regrowth in animals that should never have seen again. Sinclair called it a permanent reset. The cells, he said, were restoring the epigenetic information they had when they were young. Which raises a question. If aging is just lost information and the information can be restored, why isn't there a pill? But that is where the entire field starts to fall apart. Because for the last 2 decades, an entire industry has been telling you the answer is already here. They point to places like Okinawa, where centenarians are said to thrive on sweet potatoes. They point to Sardinia, where shepherds walk into their 110th year. They point to Icaria, the Greek island where, according to the brochures, people forget to die. These places are called the Blue Zones. They have launched cookbooks, Netflix specials, retirement communities, and a billion-dollar wellness industry. But in 2024, an Australian demographer named Saul Justin Newman published a paper that quietly tore the framework apart. He looked at the actual birth records. In the United States, 0% of people who claim to have lived past 110 have a birth certificate. In Greece, an audit in 2012 found that 72% of registered centenarians were already dead, still collecting pensions. In Italy, 30,000 pensioners were discovered to be deceased and still cashing checks. In Costa Rica's so-called Nicoya Blue Zone, after the government corrected for errors, the region shrank by 90%. Old-age life expectancy plummeted from world-leading to nearly the bottom of the global pack. And in Japan, in 2010, the government discovered that over 230,000 of its registered centenarians were either missing, imaginary, the result of clerical errors, or actually, quietly, dead. There was a man in Tokyo named Sojin Kato. On paper, he was the city's oldest living male. In 2010, police found his mummified body in his bedroom. His family had been collecting his pension for over 30 years. Newman accepted the Ig Nobel Prize for this work. His acceptance speech was 30 seconds long. The secret to extreme longevity, he said, was to move where birth certificates are rare, teach your kids pension fraud, and start lying. Afterward, a reporter asked him what he would actually recommend if someone genuinely wanted to live as long as possible. His answer? Don't buy anything. Listen to your doctor, exercise, don't smoke, don't drink. That's it. A $10 billion longevity industry, summarized in 23 words. The cleanest correlation in the Blue Zone's data is not vegetables. It is not community. It is missing paperwork. But the science itself is real. That is what makes this so hard. In 1993, the geneticist Cynthia Kenyon at the University of California, San Francisco, discovered that a single mutation in one gene, DAF2, more than doubled the lifespan of the worm C. elegans. One gene, twice the life. In 2012, Maria Blasco at the National Cancer Research Center in Madrid injected old mice with a single dose of telomerase gene therapy. Their median lifespan extended by 24%, no increase in cancer. In 2009, a team at the National Institute on Aging fed rapamycin, a drug originally extracted from soil bacteria on Easter Island, to mice that were already old. Mice equivalent in age to a 60-year-old human. They lived 9 to 14% longer. At higher doses, the lifespan extension reached 26%. To this day, it is the only pharmacological intervention that has reliably extended the lifespan of a mammal. In the calorie trial at Duke University, 220 healthy adults restricted their calories by 11.9% for 2 years. Their biological aging pace, measured by an epigenetic clock called Dunedin pace, measurably slowed. And the money is real too. In 2022, a company called Altos Labs raised $3 billion in a single seed round, the largest in the history of biotech, backed by Jeff Bezos and Yuri Milner, staffed by Nobel laureates, with the explicit goal of reverse aging the human body. Sam Altman's longevity startup, Retro Biosciences, is now valued at $5 billion and aiming for a drug by the end of the decade. Google has had its own longevity company since 2013. It is called Calico, after the California Life Company. 12 years in, it has yet to bring a single anti-aging drug to market. The pathways are real, the biology works, the money is here. So why is no one curing aging? Because every clean lab result, sooner or later, hits a wall. Sometimes the wall is cancer. Cellular immortality, it turns out, has a name we already know. It is called cancer. The HeLa cell line was taken without consent from a woman named Henrietta Lacks in 1951. She died 10 months later of an aggressive cervical tumor. The cells that killed her did not. They are still dividing today, in every continent, in tens of thousands of laboratories, weighing collectively more than 50 Henrietta Lackses. The same molecular machinery that lets a cell live forever is the one we spend $1 trillion a year trying to stop. Sometimes the wall is the science itself. In 2013, a Harvard team announced GDF11 as the long-sought rejuvenation factor in young blood. 2 years later, Novartis showed the measurement was wrong. GDF11 actually increases with age and impairs muscle regeneration, the opposite of the headline. 5 years before that, GlaxoSmithKline had paid $720 million to acquire Certris Pharmaceuticals, the company David Sinclair co-founded around resveratrol, the molecule in red wine. By 2010, Pfizer showed the entire mechanism was an artifact. The cells had not been activated, the fluorescent dye had. And sometimes the wall is the people themselves. In March of 2024, David Sinclair resigned from the presidency of the Academy for Health and Lifespan Research. The reason? A company he co-founded had promoted a supplement as the first proven to reverse aging in dogs. The data did not support the claim. The next president of the Academy, Nir Barzilai of Albert Einstein College of Medicine, told the Wall Street Journal, "The data is not good, you're calling it the wrong thing, and then you're selling it. The selling is a step too far." In 2015, a biotech CEO named Liz Parrish flew to Colombia. She injected herself with 2 experimental gene therapies and announced her own age reversal in a press release. Her own scientific advisor resigned the week she came back. A decade later, the data still has not appeared in any peer-reviewed journal. Then there is the most public case study in this entire field, a man named Brian Johnson. He sold a payments company called Braintree to PayPal for $800 million and decided to spend the rest of his life on a single experiment. He calls it Blueprint. It costs him roughly $2 million a year, a team of 30 doctors, 108 biomarkers in his blood and urine, 50 pills a day, in bed by 8:30, 3 meals before noon. He has not eaten dinner in over 1,000 consecutive nights. In 2023, he ran a 3-generation plasma swap. He drew plasma from his teenage son. He put it into his own veins, his own plasma, the blood his son had been made from. He then transferred into his elderly father, the man whose blood once made him. After 6 exchanges, he saw no measurable benefit. He quietly stopped. In January of 2025, Netflix released a documentary about him called Don't Die. In it, Vadim Gladyshev, a Harvard professor of medicine, one of the most cited longevity researchers alive, looks directly at the camera after watching the supplements, the diet, the son's plasma, the 100 biomarkers, and says, of the entire Blueprint project, "It's not science, it's just attention. $2 million a year. And here is the part nobody talks about. Every supplement at the center of this industry has a footnote next to it. NMN, the precursor to a cellular fuel called NAD, raises NAD levels in your blood and may help certain enzymes repair DNA. Maybe. The cleanest human data shows it works in cells in a dish. Whether it extends a single human life by a single day, no one can yet say. Resveratrol, the molecule in red wine, the most hyped longevity compound of the past 2 decades, turned out to activate not the cells, but the dye. Metformin, a diabetes drug, appears to extend the lifespan of diabetic patients past that of their non-diabetic peers, which would be a miracle, except that it may also blunt the gains you get from exercise. Rapamycin remains the only molecule that has reliably extended the lifespan of a mammal. It is also an immunosuppressant. Take it for long enough, and your immune system stops killing the things that are trying to kill you. And then there was taurine. In 2023, the journal Science published a major paper showing that taurine declines with age in mice, monkeys, and humans, and that supplementing it extended lifespan. In June of 2025, the same journal published a reanalysis. Taurine does not decline with age. The original premise was wrong. The author of the first paper later told a reporter that an executive at one of the largest pharmaceutical companies in the United States had approached him asking for advice because he was personally consuming 14 Red Bulls worth of taurine every day and wanted to know if he should take more. The longevity field looks like this, season after season. A breakthrough, a retraction, a breakthrough, a reversal. The Certris saga, the GDF11 saga, the taurine saga, the dog aging supplement saga. Because aging is not a single problem with a single fix, it is 12 interlocking failure modes. And every drug that targets one of them either raises the risk of cancer on the other side, or fails to replicate, or works in a mouse and fails in a human. There is one more reason no one has stopped aging, and it might be the deepest reason of all. In 1957, an evolutionary biologist named George C. Williams published a short paper called Pleiotropy, Natural Selection, and the Evolution of Senescence. His argument was simple. The genes that make you strong, fertile, and competitive at 20 are not the same genes that would keep you alive at 120. And evolution does not care whether you survive to 120. It only cares whether you survive long enough to reproduce. Aging isn't a bug. It is the receipt for being built for a life that ends before it asks too many questions. The naked mole rat got around it by evolving a giant version of the molecule hyaluronic that throttles its own cell division. The Greenland shark got around it by living in near-freezing water with a metabolism slower than continental drift. The hydra got around it by abandoning the body plan we evolved. We cannot borrow any of those without becoming something else. And maybe that is the quiet answer behind every failed pill, every embarrassed press release, every fraudulent centenarian still collecting a pension in Greece. The reason no one has stopped aging is not a lack of money. It is not a lack of brilliance. It is that the thing we are trying to stop is what built the person trying to stop it. In Italy, there was a boy named Sammy Basso. He was born with Hutchinson-Gilford progeria syndrome, a single mutation that aged him roughly 10 times faster than normal. By his early 20s, he had the body of a man in his 80s. In 2019, he co-authored a paper in the journal Nature Medicine on a CRISPR therapy aimed at curing his own disease. He died in October of 2024, at the age of 28. He had outlived almost every other child with his condition. He had spent his last decade writing the scientific blueprint for his own cure. He didn't finish in time, but he made it more possible for the children who came after him. And that, in the end, may be the only kind of immortality biology has ever offered any of us. Not the body that lasts forever, the work that does.